Great. Welcome everybody to our first session. Online people, please ask questions in the chat. I think we're doing chat, not Q&A. So thank you so much for joining us. I'd like to introduce our first speaker, Afrouz Razi.
Yes.
From Johns Hopkins University.
Thank you so much. Hi, everybody. My name is Afrouz Razi, and I'm a PhD candidate at Johns Hopkins University School of Medicine. And I'm very excited to present to you our recent work on genotype prediction of over 300,000 samples from public expression data. I always thought that this should be easy. It's not. Okay, thank you so much. So I'm going to start with a little bit of a background on Recount3 repository, as it is the basis of my project. As we all might know, advances in high-throughput sequencing technologies have resulted in production of large amounts of next-generation sequencing data deposited to public repositories. There have been a lot of efforts to provide public access to existing raw RNA sequencing data, and it was intended to facilitate reproducibility and reusability of the data. However, in practice, it is not feasible, since if you want to process hundreds of samples, it would be time-consuming and resource demanding. Previous efforts by our lab in collaborations with others have resulted in Recount3 repository by aggregating and uniformly processing RNA-seq data. Right now, Recount3 has data on over 300,000 human samples and more than 400,000 mouse samples. The available projects on Recount3 are Genotype tissue expression, TCGTEX, as well as TCGA and SRA. Recount3 is a bioconductor package, and users can query samples and studies, and they can access the data as a form of a big-wig coverage and gene or exon level expression summaries. I'm going to switch gears to our main project, which is genotype calling from this repository. As I mentioned, Recount3 has samples from different studies, and most of the samples do not have the genotype information. This would be very beneficial if you had the genotype information, for example, if you want to do EQTL analysis. Currently, the most popular tools for variant calling from RNA-seq data is from GATK software, and it's called haplotype caller. However, it requires the FASTQ files, which is missing from Recount3 due to data storage constraints. Therefore, there was a need to develop a new genotype calling model. This is a simple schematic of the data that I used for genotype calling. So if you imagine that the white squares are the base pairs and the pink squares are the read mapping to each allele. For example, for the first one, we have one read count for allele C, which is our reference, and 2 counts for allele G, which is our alternative here. With this reference counts and alternative counts in a coding region, we can then transform the data to a log ratio, which is M here, and mean average, which is S here. In our training data set, we can use this data to create these decision boundaries for the 3 different genotypes, AA being our homozygous reference, AB being our heterozygous, and BB being our homozygous alternative. In a new data set, when we have the reference counts and alternative counts, we can then predict the genotype based on where they end up in these decision boundaries. We evaluated our prediction accuracy by looking at GTEX data set, as well as GeoBodies as an out-of-study test set. If you only focus on the overall and the black bars, And we get an overall mean accuracy of very high accuracy of 99.5 for GTEx and 99.2 for geobodies. We were interested to see if we have a toy model that we always predict homozygous major for all the samples, what would our performance be? And we surprisingly, we saw a very high overall accuracy of 95.7. 95.7. This was surprising, and this shows that overall accuracy is not a good measure to report the accuracy, because you never get the minor allele correctly. So here we show that should show accuracy for major and as well as the minor allele for both of the studies, and we see consistently high prediction accuracy for both of them. Furthermore, we were interested in seeing if our prediction accuracy changes across the tissues. So we use the 54 tissues across the GTEx, and each dot here represents a tissue, and we saw a consistently high prediction accuracy for all of them, for both major and minor. We determined that our model performance is highly dependent on coverage as well as minor allele frequency. Here I'm showing a GTEx test set as well as geobodies on each row and minor allele frequency on each column. The coverage is shown on the x-axis and the prediction accuracy is shown on the y-axis. And as you can see, as the coverage increases in all the bins for the minor allele frequency, you get higher prediction accuracy. We use this model to have an expected prediction accuracy for each genotype that we present to the user. Lastly, we wanted to see if our predicted genotype contains any biological signals. Therefore, we used 1,000 genome DNA sequencing data to generate a PCA plot for as a reference, because we had available data for 5 different superpopulations. Before applying this to other data sets, we wanted to see if we can use the predicted genotypes from RNA-seq data to correctly predict the population for each sample. Therefore, we use the geobody samples since we already had the populations for each sample. And we saw that each sample correctly grouped with the corresponding population. We used k-mean clustering to assign samples to each superpopulation for the rest of the samples in recount 3. And on the right, I'm showing the population composition of recount 3 based on the assay type. In summary, we developed a statistical model to predict genotype from reference and alternative counts in recount 3. Our model has high prediction accuracy regardless of tissue or study, and using predicted genotypes, we can infer the population structure of recount 3. The data is available for processed RNA-seq data through recount 3 package in Bioconductor. The genotype information will be available, but we are currently working on the privacy protection plan. So hopefully in the next month, you will hear about this. The genotyping pipeline is right now as a form of a GitHub package, and a paper is about to be published hopefully in the next month, so look for this title. I want to acknowledge the work that was done by Chris Liu and thank all the people who were involved in this project. And I would like to thank the Bioconductor committee for giving me this opportunity to present. Thank you so much.
We are going to go to the next speaker. We're going to have all of the talks at once. And I was wrong for the virtual people. We are supposed to put your questions into the Q&A because people can upvote your questions. All right, so our next speaker, Pedro Bologna. Okay, let me get your slides up. Oh, where is the... If I can find it. Oh, where's the drive? Okay, the second one. Now I need to share it. Okay, that should work.
All right. All right. Thanks everyone for attending this section with short talks on RNA-seq. I'm Pedro. I'm a postdoc at the WIHI in Australia. And today I'm going to briefly introduce Catch Salmon and Catch Callisto, two functions that are in the EDGAR package for people to analyze differential transcript expression within our EDGAR framework. So in the past 14 years, most of the analysis of RNA-seq data has been focused on gene-level inferences. But since the advent of fast quantification tools such as Salmon and Callisto, analysis of RNA-seq data at the transcript level is becoming more and more popular. To give you an estimated total read count per transcript, these tools will handle a missing data problem and probabilistic assign reads to transcripts. And this comes with a major new source of variation that is due to the high levels of sequence similarity among transcript software annotation. What Catch Salmon and Catch Callisto will do is to estimate that extra source of variation and provide ways users can analyze transcript level data within EDGAR. So it's important to realize that mapping ambiguity is an extra source of variation in analysis of RNA-seq data at the transcript level. So let's suppose we have a set of samples for which you're going to run our RNA-seq experiment. We have a certain gene with an underlying abundance pi that we're going to sequence fragments of its cDNA molecules up to a library size to get total gene counts. Well, it turns out that the Poisson variation that describes quite well this process of sampling mRNA molecules and counting genes, gene level counts, does not apply for transcript level data. And that's mainly because there is this mapping uncertainty associated with the quantification of transcript expression. You can easily see that the assumptions that we make for gene level analysis do not apply for transcript level data. If you look at, for example, mean variance relationship plots from any real RNA-seq data set that you might have at hand. While the Poisson gamma model or the negative binomial distribution fits well the data at the gene level, we cannot say the same for data summarized at the transcript level. So actually, mapping ambiguity increases this technical variation for transcript quantification. And we did a quick exercise of simulating technical replicates, which you can access from the page listed at the bottom of these slides. And what we saw was that quantified counts from transcript coming from multi-transcript genes had an associated multiplicative increase in their observed variance after quantification in comparison to the Poisson variation found for single transcript genes. To account for this effect, we propose to use a quasi-Poisson model for the conditional variance of transcript counts, where mapping ambiguity can play a role in the technical variation via this mapping ambiguity over dispersion parameter sigma square, with transcripts coming from multi-transcripts genes having an associated variance that is greater than that of a Poisson model. Well, that results in a variance function for transcript counts that's quite complicated to work with because mapping ambiguity can be very transcript specific, and biological replicates only allows us to estimate certain parameters such as the mean count and the negative binomial dispersions. So in EDGAR, we tackle the problem of DTE, differential transcript expression, by providing precise estimates of that mapping ambiguity over dispersion and calculating effective transcript counts. These functions, Catch Salmon and Catch Callisto, they will take output from either Salmon or Callisto, respectively, in particular, the total read count per transcript and per sample matrices of bootstrap resamples. The idea of our approach is that we can use these bootstrap resamples to estimate the extra variation that is mainly due to mapping ambiguity. We propose to use a moment estimator to estimate this extra to Poisson over dispersion, followed by some empirical based moderation. And then we would divide the counts by those estimated values to obtain effective transcript counts. The interesting thing about this approach is that effective transcript counts really reflect their precision, and we end up with a variance function that is equal to the one from a negative binomial distribution. That means that we can apply any gene level method to analyze transcript level data while preserving fold changes and estimating sources of technical and biological variation that are free of mapping ambiguity. This approach can be visually appreciated via biological coefficient of variation plots, in which we show the estimated square root of the negative binomial dispersions as a function of the transcript abundances. And immediately we can clearly see that these estimated dispersions can be artificially high for certain transcripts that are associated with high mapping ambiguity. But upon count scaling, these effective counts really reflect their precision with the biological variation that is representative of common RNA-seq data sets. Then our favorite differential transcript expression pipeline would involve processing your data sets with Salmon and importing those results with AGR. You would pretty much only add 2 extra line of codes to your standard analysis with AGR, first by importing the results with Catch Salmon and then dividing the counts by the mapping ambiguity that was estimated. And then you can proceed the analysis as usual with our quasi-likelihood pipeline and F-tests to assess differential expression. We did an extensive simulation study that you can get more information about, again, in that same page, to assess the performance of our approach in a controlled scenario. But basically what we did was to use the R-subread package to generate in silico sequencing reads in FASTQ format. We quantified transcript expression with Salmon, and we ran AGR with count scaling along with a set of other methods to compare performance. But basically what you can see from these 2 bar plots on the left is that we saw that AGR with count scaling had the highest sensitivity to detect differential expressed transcripts, independent regardless of the sample size. We also saw that AGR with count scaling approach had a pretty good control of the number of false discoveries, as I show on the 2 plots on the right. But basically the big impact here came in scenarios with low sample sizes, for which other methods either had low power to detect the transcripts or limited control of the false discovery rate. We also assessed the performance of our approach in all simulations to control the type 1 error rate. Those are presented on the 2 left bar plots, and you can see that pretty much all methods do a pretty good job in controlling the false positive rate, with Swish, another bioconductor method for GTE analysis, being the best here, thanks to its permutation-based tests. Regarding time, users can analyze transcript level data in just a few seconds, and that seems to scale pretty well with the number of replicates as we have more data sets to analyze. So I hope I gave an overview of Catch Salmon and Catch Callisto, how you can analyze your transcript level data with AGR, with our estimates of mapping ambiguity and count scaling approach. If you're interested in learning more, please take a look at our preprint on bioRxiv, or take a look at the AGR Users Guide that since April has a new GT case study. Last but not least, I'd like to thank my co-authors who have worked with me in this project recently, and the funding sources. Thank you.
Thank you very much. See how quickly I can get this done this time. Okay, next we have, I think, Sonalia Aurora. Are you able to share? She's a virtual presenter.
Hi, everyone. I hope you guys can hear me.
We can see you, but we don't. Can you share your slides?
Yes, just a second. Okay, I think everybody can see my slides. I'm just going to presenter mode. Is that loud enough, Jenny?
Yeah, you're fine to go. I'm going to try to make it bigger on our end here.
Okay.
that okay?
All right. Hi, everyone. Thank you so much for letting me present remotely. I work at the Fred Hutch Cancer Center, and today I want to talk to all of you about a project which we started during the pandemic, when the pandemic hit, and it just got published in March, and I'm happy to talk about it today. So briefly, CNS tumors or brain tumors are one of the most common type of cancer and one of the second leading causes of death in the United States and Canada. And I work in a brain lab, and one of the things that we started doing when the pandemic hit was we were like, oh, all the wet lab experiments kind of stopped, and we decided to take publicly available data sets and see what we could learn from those data sets. And so there are a variety of primary brain tumors, which you guys can see in the pie chart ahead. The most malignant one, or the one with the highest mortality rate, is glioblastoma, and the ones with the most benign ones are meningioma. I want to thank our first speaker, Afroz Razi, who actually introduced about RECOUNT-2. At that time, RECOUNT-3 was not available, so all my data analysis is based on RECOUNT-2. So we decided to look up publicly available data sets where we could find different brain tumors. And RECOUNT-2 was a great source because they had taken all the data, bulk RNA-seq data, and reprocessed it from scratch and kind of made it available for us to use. So we actually got the glioma data sets from TCGA. There are two different brain tumors in TCGA, the low-grade gliomas, which are less aggressive, and the GBMs, which are more aggressive. We also took, there's a database called the Chinese glioma, which was also present in RECOUNT-2. We took that. And then as a comparator, we decided to take normal brain from GTEx. So about 1,400 samples to kind of study the differences between normal brain versus adult glioma. Pediatric tumors are also present, and there are a wide variety of them, with about 29 different types of pediatric tumors. We found this in a repository called CPTN. And so from there, we directly downloaded the data from them. That was not available in RECOUNT-2. And then we took the RNA-seq, bulk RNA-seq data from each one of them. We selected the samples from the studies. So basically, TCGA contains 33 types of tumors. We just took the brain tumors. And then we normalized it to transcripts per million, TPM normalization. We did some batch correction using SVA package, which is present in Bioconductor. And then we constructed a UMAP, which we call the brain UMAP in the lab. And then we decided to overlay it on different types of genomic data and do some 3D visualization. And obviously, this was to help the clinicians and people who don't really code, help them understand big data, and can you visualize it and make it intuitive in a format that they can actually access it. So this is what the brain UMAP looked like. What you can see is dimension reduction was applied to all the bulk RNA-seq data sets. Each point here is a patient. The green data sets that you can see on my screen here are actually normal brain, so they divided into two clusters. What you see in the middle, in the blue, is actually the pediatric tumors, and then orange, pink, and purple are the adult gliomas, which are coming from 3 different data sets: the TCGA lower grade ones, the less aggressive ones, and the Chinese glioma data sets. And so obviously we put in a certain amount of consideration about which dimension reduction technique we should use. So we looked at PCA, we looked at TISNE, and we looked at UMAP. And after a lot of discussion with the neurosurgeons and the clinicians on our team, we decided that the UMAP best captured the clustering of the data and the groups more as it fit in with the understanding of the disease to date. And so this is the brain UMAP, which I showed in the previous slide, where everything is colored in by the data source, but you can actually then go in and color it by the different tumor types. So the adult gliomas can be divided into 3 different categories. The red ones are the most aggressive ones, the astrocytomas are slightly less aggressive, and the green ones are the least aggressive ones. And then the pediatric tumors, as I was telling you, there are 29 different types, and you can see that they form clusters individually of their own different sets. So one thing that we noticed is if you just look at the adult healthy brain from glioma, the bulk RNA-seq data was able to capture the different regions of the brain correctly. So the neurosurgeons in our group were really excited by this because you see this petal kind of shaped structure. It actually segregated into 3 different petals, which actually work very well with the way the regions of the brain are arranged in the structure. And so the cerebellum, which is this other second thing right on top, or what you see in dark green, completely forms different structure. And so they were very happy to see that the different regions of the brain can have different bulk RNA-seq data as well. And so one thing we wanted to explore was, how well do these data sets merge? And I think this is a shout out to the Recount team. Because they analyzed everything from scratch, they took the FASTQ files and realigned everything using the same pipeline, we could see amazing co-localization of these 2 data sets. So the 2 data sets that I'm talking about is TCGA, which is primarily patients in North America, CGGA, which is Chinese glioblastoma patients in collected all over China. And then these patients were sequenced almost 10 years apart. So there was a lot of differences in, when the patient samples were collected, how they were collected, but they show extremely well co-localization. From both these repositories, we had a lot of different information. So for example, we had the grade, right? And so the brain tumors are divided into grade 2, grade 3, and grade 4. And you can see that the grade 4 kind of co-localized in the same area. On top, I'm showing the TCGA data set with the CGGA colored out, and at the bottom, I'm showing the reverse. And you can see that this part of the UMAP, all the grade 4s were there, the grade 2 and grade 3s were at the bottom. If you look at the IDH mutation status, that's something else that clinicians look at in the field, and the aggressive ones are supposed to have the mutation in it. And so you can see that, those also localize in one particular area of the UMAP. Same goes with the copy number, 1P19Q code-deleted status. They also localize in a certain region of the UMAP. So our UMAP was able to capture the different ideas of the disease or the different characteristics of the disease, which clinicians already know. And so that was really exciting and a good validation for us. We can also use this, to understand overlay the clinical information that we have from these data sets and understand it better. So these are the 3, again, looking just at the adult glioma subtypes. You could overlay the years on it, and then you could see that patients who were diagnosed at a much higher age with a much aggressive disease formed like a region. The males and females, like if you color the UMAP by that was equally dispersed. There was no particular region in which they were dying. DNA methylation. So DNA methylation data, we did not collect it, but we just overlaid the DNA methylation data from these particular patients. And you could see that even that formed distinct clusters. So the RNA-seq clustering and the DNA methylation subtypes really agreed well. We could overlay chromosome status on these on this UMAP as well, and saw that also agreed with what the clinicians know. The mutation status. So all these things were really good validation to ensure that our UMAP was actually agreeing with what the clinicians thought, and it was really good for them to understand, like, okay, this is where the patients with mutation status, copy number changes sits in this area of the UMAP. So next, we also wanted to look at the pediatric ones. And so medulloblastoma, this is like a benign kind of tumor, mostly seen in children. And the interesting part about this tumor is that usually to understand the subtype of it, people have to actually send the sample for a methylation analysis. So this was the first time that we could take the RNA-seq data and see beautiful clusters of it. So ideally, if RNA-seq data was collected on this, subtyping or characterization of the subtype of the tumor could be figured out with RNA-seq in addition to methylation as well. And so you can see the different groups kind of coming out beautifully in this sum. And these guys, which are lying in the rest of the UMAP, are probably misdiagnosis based on what the neurosurgeon in our group says. So that's great. So you can characterize the data set, you can figure out clinical information, but then you can also use it to visualize gene expression trends. So for example, this is a particular pathway, all the genes that belong to the reactor mismatch repair pathway, and you can see that in all of them, like the expression of the genes go from 0 to 10, 15 based on the normalization method. And you can see it's mostly upregulated in adult glioma. All of them are also upregulated in medulloblastoma, the subtype that we were looking at, except for these 3 genes. So for example, if somebody was studying this pathway and wanted to understand how these genes work, you could figure out how the gene behaves in a normal brain versus pediatric tumors versus adult brain. Right, and so this is also something that can be really helpful to people who are designing experiments. And just last 2 slides, you can also visualize different pathways. So we did a GSV analysis and figured out pathways on it. And then finally, just to recap.
Sonali, you're over time. Sorry.
Sorry. Yes.
Can you finish up?
Yes. Thank you all for presenting, and then I'm just going to leave it at the slide and.
Okay, great. Can you stop your share? Maybe I can.
Oh yes. Sorry about that. So thank you everyone, and yeah, I'll be available in the chat if you have questions.
Great. Okay. Sure. Okay, so our last speaker is Diana Gonzalez Padilla, right? And okay. Good. Good.
Okay, well, hi, I'm Diana Gonzalez, and today I will be talking about the project I've been working on during the last year, in which we're trying to identify what are the effects of nicotine and smoking exposure on the developing brain of mice. So just a little about me, I'm an undergraduate student of genomic sciences in Mexico, and right now I'm a member of the data science team at the Liver Institute for Brain Development. Overall, I'm interested in pharmacogenomics, drug discovery and development, transcriptomics, and all that apply to the study of the brain. So just a little about the smoking mouse project. As you know, the maternal smoking during pregnancy is a major health concern in the world, not only because it has an impact on the smokers, but also because it affects the health of the offspring, particularly in their cognitive and behavioral traits. But one important question that is kind of ignored is if those effects are caused by the cigarette smoke only, or if those alterations could be driven by specific components of the cigarette, such as nicotine. So that's the question, and the problem is that most of the studies have focused on analyzing what are the effects of the smoking exposure only, ignoring the role of the nicotine that is an active component of the cigarettes. So in order to address that question, the researchers of the Liver Institute for Brain Development took adult mice, all of them were female, and some of them were pregnant and the rest were not. So a part of these adult mice were administered nicotine directly. The rest were controls for the nicotine experiment. Then there were these mice that were exposed to cigarette smoke directly in chambers, and the rest were the controls for the smoking experiment. And pups were born to all these pregnant mice, either nicotine exposure controls or smoking exposure controls. And brain samples were taken from the developing brain of the pups that were born and also from the adults. And also, as you can see, there were blood samples from the adult mice that were part of the smoking experiment. So in total, there were 208 samples. RNA was extracted in sequence, and the expression data was generated at the gene, transcript, exon, and junction level. So what were the main goals of the project? Initially, the first question, the most important one, was what are the effects of nicotine and smoking individually, right? That is, what are the affected features, either genes, transcripts, exons, and junctions. And once we have that information, to compare what are the effects of nicotine versus smoking, and that in the developing brain of the pups. And as secondary objectives, we did the same analysis, but for the adults, comparing the affected features in nicotine exposed versus controls and smoking exposed versus controls. And with that, we compared the results in adults with the results in the pups. And then we have these blood samples for adult mice that were part of the smoking experiment, because there are these epidemiological studies claiming that it is possible to capture the environmental effects such as smoking exposure on the brain by analyzing the blood. So we tried to identify if there were any common biomarkers in brain and blood, that is, like if there were common differentially expressed genes in both tissues. And finally, once we have the result in mice to compare, of course, we want to extrapolate the result with human. So we took the results of 2 previous studies in human and compare our results in mice. So this is a summary of the workflow. What I did was basically a differential expression analysis. Of course, there was this step in which I have to prepare the data to normalize and filter it. And I also separate samples by tissue, age, and experiment, because the biological questions we were interested in were different. Then I did exploratory data analysis. I assessed the quality of the samples. I explored the sample and gene level effects. Then I performed the differential expression analysis itself, in which I try with different models, and I compare the differentially expressed features in different sample groups. And with that, I performed the gene ontology and enrichment analysis. I visualized expression patterns of differentially expressed genes in heat maps. And since I have also the information at the junction level, I was able to annotate which were the preceding nearest and following differentially expressed, the preceding nearest and following genes of the differentially expressed novel junctions that were completely unknown. So these were the results. In pups, there were more than 1,000 differentially expressed genes for the nicotine experiment, but this is just for the developing brain of the pups. And as you can see, there are discrete patterns of upregulated and downregulated genes. They were also differentially expressed transcripts and exons. So in these plots, we are comparing the T-stats of the, We're comparing the T-stats of the transcripts and the exons versus the T-stats of their genes. So we can see these nice correlations between them. And in orange, for example, each dot corresponds to a transcript. So in this case, the orange ones are differentially expressed transcripts that come from non-significant genes, just to present how enriching it is to include information at the transcript level. So for example, this transcript comes from the gene BCL11A that is implicated in autism and schizophrenia, and it has a transcript that is significant. And there are these red dots that correspond to transcripts that come from the same gene, the ones that I'm highlighting that are circled. And they come from the gene PNSIR that is predicted to be active in present active zones. And what's interesting of these transcripts is that the gene was differentially expressed, and it also has one upregulated transcript and another that was downregulated. And in the Venn diagram, we can also observe that there were differentially expressed genes that have also differentially expressed transcripts and exons, which presents how consistent the results were at the different levels. And then for the smoking experiment in the developing brain of the pups, again, there were more than 4,000 differentially expressed genes. We can observe the upregulated and downregulated genes in their expression patterns. And there were thousands of differentially expressed transcripts and exons again, and an overlap between the different features. They were, for example, this transcript from the gene BTF3 that is implicated in utero embryonic development and was significant. And these 2 other transcripts, one was upregulated and the second was downregulated for the same gene, more for L2, that it has a BA's expression in placenta and the brain. If you remember, the second main objective was to compare the results of nicotine versus smoking. So in this table, I'm summarizing the number of differentially expressed genes that were either upregulated or downregulated in either the nicotine or smoking experiment. And in different colors, after the analysis of the gene ontology and cake enrichment analysis, we can see some interesting functions related with the brain. So in orange, we have more than 1,000 genes that were upregulated in smoking, but were not significant in nicotine, that are implicated in the SNR complex. Then we have in red these genes that were upregulated only in nicotine, but not in smoking, that have to do with the paraminergic synapses and long-term depression. And interestingly, there were 47 genes that were upregulated in nicotine and downregulated in smoking. And they codify for these proteins with heat-shock protein binding activity. And there were 17 genes that were downregulated in nicotine and upregulated in smoking, that have to do with SNR complex and SNR interactions. And the results of a secondary analysis, they were not differentially expressed genes in brain, probably because it is complicated or not that simple to model the effect of nicotine and smoking exposures. They were also not differentially expressed genes for the smoking exposure in the blood, but they were mouse and human brain differentially expressed genes that replicated in mouse blood genes. And also with comparing with previous results in human, they were both adult and post mouse brain genes that replicated in both human prenatal and adult brain and vice versa. And there was this gene NRCAM that was differentially expressed in both the prenatal human brain and also in the poop brain, both of them exposed to cigarette smoke. And this other gene Marco that was differentially expressed in human adult brain and also in the nicotine exposed brain. And some of the differentially expressed genes that I found in mice were also found to be associated with tobacco use disorder in a GWAS study performed in human. So as you can see, we have plenty of data. That's the reason why we decided to create a smoking mouse package that offer access to the raw data that I use for my own project, and also it contains the results of my own analysis of differential expression, and you can use it to answer many questions related to smoking and nicotine exposures on either the brain or blood of mice. And it also offers access to human data from SMIC in 2018. So this is the people that I would like to thank, and yeah. Thank you.
Okay, great. Thank you to all our speakers. We are a little bit over time, and we can try to take one or two questions. Any in the room?
Question for Pedro. Okay, so very, very exciting work. And I was wondering, I saw, so Matt Ritchie's on project.
Yep.
Have you explored the importance of your methods for long read, which also has there's some fragmented reads that matter?
Do you want to come up here?
Yeah. Yeah, thanks, Mike. Good question. Yeah, we tested our approach with long reads. We processed long reads with salmon, and we saw, as we expect, no mapping ambiguity for long read data. So our estimated values of sigma square for that parameter, it's pretty much very flat, close to 1. Yeah, that's what we did. Yeah.
So we have a question from Yining Pan online for the Recount 3 data set. Specifically, are the human samples in Recount 3 general population, do they have clinical information associated with them? That struck me as one for her.
Yes, thank you so much for that question. That's actually very helpful if you're looking at different diseases. Most of the samples are from healthy individuals. We do have metadata in Recount 3, but some of them are missing. And to have a complementary metadata, you have to use meta SRA, which is done by another group. But yes, you can find information from different clinical, you can find different clinical information from that resource.
I have a question for the catch, the catch callistical person. Very interesting work. I would like to ask, how does the catch salmon plus agile differential expression works for low expressed transcripts like long non-coding RNA isoforms? Because from my experience, they only appear on switch differential expression.
So pretty much, so when you divide, I mean, it depends on what you're interested in analyzing, right? You can focus your analysis on protein coding genes only, but you can keep your long non-coding RNAs, of course. If you want to keep them, and then you're going to divide the counts by their mapping ambiguity. If they survive filtering, after you deflate them, then you're going to keep them in your analysis as usual. But yeah, when you divide the counts by the mapping ambiguity, remember that you're bringing everything down, and chances are that you're going to remove them if the mapping ambiguity is high. Yeah. Mm.
Okay. I think we need to have time to change rooms and get new speakers up, but thank you everybody very much. If you have other questions for the speakers, please get in touch with them, find them if you're in person, otherwise ask them, you can chat with them on Hopin. Thank you very much.